So today I'm going to share some research that follows up on some of the previous work I've done on cyclical mutations. And this is, we've been working on a population dynamics simulator uh, based on the work of Lynn Caporale. So that there's a book called The Implicit Genome that has a lot of, let's, I wouldn't quite say that it is supportive of the idea of cyclical mutations. It doesn't mention that per se, but there's a lot of overlap between the concepts, and I think you'll find a lot of if you read the book, you'll find that the idea of cyclical mutations is implicitly implied in the implicit genome. So anyway, that's why it's named the Implicit Genome Simulator. And we're covering kind of a non-Darwinian view of genetics. And if you want more information about this, I've got other videos, and I'll point to them in a minute. So, kind of the goals of this presentation is, first of all, I want to briefly introduce the concept of cyclical mutations. I have other videos that go into it even deeper, but I want to at least, this is the only video you see, you can kind of have a good feel for it. I'm going to demonstrate the simulator in action, discuss the current implementation, and I'm going to kind of set the stage for our next steps and hopefully can get some feedback from anybody watching. So, all right, so that's the game plan. So let's move on to cyclical mutations. All right, so let's talk about non-Darwinian biology. Okay, so when we talk about mutations, they usually have a connotation. When someone says mutation, usually what pops to mind is someone thinks, well, random mutation. All right, they think that mutations are random and haphazard in some way. Uh, well, the problem here is that it's the way that I think about it is like bacteria. When we first discovered bacteria, we viewed bacteria as being entirely negative, and the reason for that is because we could really only identify bacteria based on its negative effects on people. And so we just kind of assumed that they're all bad. And that actually led to some issues where people were trying to avoid bacteria at all costs. But it turns out that bacteria are actually important to a lot of physiological processes. We just only saw the ones that were bad, and the bad bacteria are only like maybe 1% of the bacteria available. So what if the same thing was true about mutations? What if mutation itself was a physiological process and that just happened to go bad sometimes, but on the whole, it was actually an interesting and necessary process that is not haphazard at all, but actually there is a design and pattern to it. So, what if mutations weren't random and haphazard? What if they were designed? What if they were part of a standard physiological process? Okay, so we can talk about mutations, and just like we have a system for what keeps the heart beating right, and we have a system for what causes the brain to function, what if we have a system for what causes mutations? What would that look like? And so what I've argued in other videos is that this would lead to the idea that there are cyclical mutations. So if you think about it, for random mutations, let's just say you have if there's an organism that has 1 billion base pairs, right? Well, if you have a mutation, then you basically have essentially a 1 in 1 billion chance for that organism's line to get a reversion mutation. So you're not really it's not really all that likely to occur. It kind of can be considered a rounding error, and by the time it does occur, the context is probably sufficiently different that um, it's not even really a back mutation per se because it doesn't restore previous functionality. So that's kind of the way that we think about mutations in general is we think about them kind of being cumulative. And most population dynamic simulators, uh, they kind of do this where they kind of have kind of an origination and fixation model. So you have a new mutation pop up out of nowhere. It gets assigned a fitness value, and then it sticks around. Um, but what if instead the mutation process was designed? What would that? How would that change the way that we looked at things? So anyway, so as I said, we typically think about mutations being directional. So if we think about cyclical mutations, let's say, what if there was a predetermined range of modifications around which most mutations occurred? What if there was kind of predefined ways that genes were likely to mutate, right? Um, so probably the best thing to do here is just to give you some example mechanisms that we know about to kind of get your mind seated for how these might occur. One of my favorite examples is organisms that have fixed alternative genes encoded in pseudogenes. So this has been found in Neisseria, for example, where they have an outer coat protein called pilin, and there's one expression locus for pilin, but they have numerous alternative codes for the pilin gene, but they are in pseudogenes. And so what happens is periodically there will be a mutation such that one of the other alternative codes will be copied into the expression locus. And the mutation is always one way, it's always copied from the pseudogene into the expression locus. So it never loses any of its alternatives, it just switches back and forth between them. Okay. So that's one. So you basically have distinct alternative copies in your genome of alternate coding sequences for a gene or a gene component. You also have phase variable genes. These are genes whose configuration basically flop back and forth between two alternatives. Usually it's turning them on and off. One common technique is DNA inversion, where a little piece of DNA will be inverted, turning it off, and then it'll get reinverted again, turning it back on. Another interesting one is simple sequence repeats, and so these have been likened to tuning knobs in the genome. So simple sequence repeat is a sequence that has just usually 1 to 9 base pairs, and they just repeat over and over and over and over again in the genome. And so it's a simple sequence and it repeats. Now the interesting thing about these is that they are a lot more mutable than pretty much any other site in the genome, but they pretty much only mutate by adding and removing copies. So they get longer and shorter. So they basically uh, act as tuning knobs in the genome. So for example, this is what determines the bend in the dog's nose is these how many simple sequence repeats these have. So this allows for biological tuning knobs to exist in the genome. Then you also have insertion sequences and excision enhancers. So a lot of organisms have these insertion sequences that are actually sequences that are coded for by the genome that insert themselves in other locations. And then they have excision sequences, which basically they look for the markers at the end of these and pull them out. So you have a cycle of insertion and removal, and insertion and removal. Um, and so in a lot of organisms, mutations which are potentially cyclical, which fall in one of these categories, they actually far outweigh directional mutations such as single nucleotide polymorphisms, sometimes by a large amount. And I mean, a lot of times, each of these individual classes outweigh single nucleotide polymorphisms. Um, so I find that really fascinating. Now, so what we're going to be modeling here is just cyclical mutations. Now, I think that real population dynamics has a lot of other dynamics associated with it. Obviously, there are accidental mutations, and I'm not taking that into account at the moment. There are also, I think there's a combination of cyclical and directional mutations. So a way to think about this is if you think about an insertion sequence, where the insertion sequence itself has a simple sequence repeat attached. So the element can be inserted, and then from that insertion, other cyclical mutations can be applied on top of that. So those sorts of things happen quite a bit. We're just the purpose of the current research is just to look at the first order consequences of having basic cyclical mutations. So there's obviously more downstream work we could do, but we're focused on. I think that cyclical mutations is a mode that has not been explored properly. So exploring that on its own, I think, is an important step forward. So now there's a problem of deleterious mutations. So you say, if all these different options are physiological designs, why are so many of them deleterious? So, for example, the beneficial deleterious ratio of mutations is really, really low. So there's very few beneficial mutations for every deleterious mutation. So why is that? Um, and I think the real reason is that there's actually two non-equivalent meanings of deleterious. Cordova talks about how there's this idea of fitness, and there's two meanings of fitness. There's the population genetics definition of fitness is basically how fast an organism reproduces, but then there's also the biochemical meaning of fitness, which is basically how well does this function, and those are two different meanings, but they're not equivalent. Failing to function does in fact impact uh, your reproductive fitness, but having a lower reproductive fitness does not necessarily mean that there's a problem per se in your physiology. So example that I like to use is, if you imagine your car, um, if there's a car engine and I just take my hammer and just bash something random in my car, well, that causes it to not run as well. Um, but another thing that my car can do is my car actually has a setting for what the idle RPM should be. And if I set the idle RPM too high or too low, it can cause the car to not run as well. But the difference is that the first one is just something that's totally outside the parameters of what a car should do. The second one, I'm basically working within the framework of what a running car should look like, and I'm tuning it. Now, I might be tuning it well or badly, but I'm tuning it according to the car's internal parameters. And so the two non-equivalent meanings of deleterious, one of them is kind of the philosophical meaning that we broke it. Another one is the empirical one, and that's just it doesn't reproduce as often. And so once we recognize that the fact that it doesn't reproduce as often does not mean that it's a broken configuration. It doesn't mean that it's actually misfunctioning. It may just be that it's a lesser tuning, right? So we can't use these as proxies for each other. So when we're thinking about this, there's a thought experiment I like to do where you just think of, imagine an organism that has a single gene that has just a fixed number of configurations. Let's say 10. So there's a single gene and it can mutate into one of 10 different configurations. And all of these, let's just say that all of these configurations are designed, right? They each one works better in a different environment, but all 10 of them were designed. Okay. So now, if you think about it, if it stays in a single environment for a long time, if it just sits there, well, whichever of those configurations is most, whichever of those configurations most matches the environment is going to predominate. Okay, that's actually what the fundamental theorem of natural selection says, that if you have a set of configurations, um, because it's an exponential growth curve, whichever configuration is most fit will dominate the population. So that has an interesting byproduct, is that because once it's dominated the population, then pretty much all mutations go from that optimal configuration to one of the other ones. So that means all the mutations from that point will be deleterious and they will be lesser fit. So despite the fact that all the mutations are designed within a specific environment, if an organism stays in that environment for a long time, most mutations are going to be deleterious. Right? Now, but there's an interesting consequence that I think that we can use to kind of uh, determine whether or not my hypothesis is correct, and that is under this system, when the environment changes, you'll have a sudden spike in beneficial mutations because all of a sudden since the reason that the beneficial deleterious rate was so low was because it had form fitted itself to the environment, and out of the configurations it knew they most matched, when you switch environments, then you're going to have, uh, since all of them are going to be coming from a less fit, a not optimal bacteria, then they're more likely to be going closer to the to the optimal rather than away from it. So So that's enough background. If you want some additional background, here's some other videos that I've done on this. You can kind of cover kind of the history of the research in this topic. I think you'll find it interesting. So, as I said, most population dynamics simulations assume mutations accumulate, but what if they cycle instead? So, one of the popular um, population dynamic simulators I'm aware of is Mendel's Accountant. I think it actually does a great job of modeling Darwinian biology. But the question is Darwinian an actual model of how biology works, um, or is there another model that we should be looking at? So that brings us to the actual simulator itself. Okay, so the popular dynamics simulator that I built, it's written in the Go programming language. I've been kind of specializing in Go because it allows you to write fast code and write it quickly and write it cleanly, and it doesn't require a lot of extra tooling. Um, you can just get up and running and started. You can find the simulator at on the GitHub page. It's command line operation, outputs to CSV, and the other thing I haven't seen this a lot in other scientific simulators, though I think they should be more prevalent. Uh, it actually provides you with the random number seed. So if you want to rerun an experiment to make sure you have reproducible data, you can specify the random number seed to use. So every time you run it, it'll output the random number seed, and you can re-input that to get the exact same results. So you can communicate that to other people. And then the code it's very easy to customize. There's not even a lot of code there. It's a pretty straightforward application. So you should be able to easily make adjustments. So here's what a sample run looks like. So if I want to run one that has 10 loci per organism, 100 iterations per environment, and 5 different environments, that's the command line you use. The output just says, um, hey, here's the seed I started with. So if I run with the same commitment number, the same arguments, I can add in a dash dash seed parameter with the given seed, and I can replicate the exact same results. And so this will run pretty quickly. The And this is the results that you get. And so you can see, remember I'm running 100 generations per environment. And so you can see on the 101st generation, we have a sudden spike in fitness, or in the beneficial deleterious ratio, not in fitness. Fitness goes down. Why is there a sudden spike in the beneficial deleterious ratio? Because uh, the organisms were becoming dominated by the most beneficial configuration. And so all of the mutations were away from that configuration once it dominated the population. But once the environment switched, that was no longer the case, and the beneficial deleterious ratio bumped way up and then slid back down. So on the right, you can see the environmental change, or the fitness change, and so you can see the fitness is gradually increasing and kind of plateauing, and then it drops down, gradually drops down, gradually increases. Um, and that one has a environmental change that leads to a fitness increase, which is definitely possible. You can have an environment that is overall more favorable. Um, but you'll notice that at each level, the beneficial, whether or not the fitness increases or decreases, the beneficial deleterious ratio always jumps on a significant environment change. And so we can find the same dynamics if we run it for 10,000 generations instead. Um, so here I run it with the same seed, so it's going to get, if you run it with the same seed and the same number of loci and the same number of environments, you'll get the same set of starting environments. And so here are basically the same environments, but we're going to run for 10,000 generations each, and you'll see basically the same dynamics. It's just there's a lot of steady state behavior after a while. So how does the code work? Well, so here's what a gene locus looks like. So in an implicit genome, each gene kind of has an implicit range of values it can mutate to. So we have kind of three types of genes we're modeling. The first is a fixed configuration gene. So a particular gene can have X number of fixed configurations. The other type of gene is a continuous change gene. So this gene can have any value between 0 and 1. So that's kind of the model SSR type behavior, so that the mutations can go through a range. And then we have on-off genes, which is basically the fixed configuration gene that has two states. And so a mutation simply moves a gene from one possible value to another. All right, so that's what our mutations do. So the way this looks like, an implicit locus, uh, we give it an ID for tracking. We give it a type, so whether it's continuous or discrete. We give it for discrete valued locus, uh, we give it a maximum of the range. And for continuous valued locus, we tell it what uh, the maximum continuous change is allowed on it. And then mutability is the chance of mutation each generation. Right now, that's fixed at 1%, which would be 0.01. So an actual locus in an organism simply points back to the implicit locus and then the value for it. So then an environment, the environment is just a mapping between gene states and fitness values for each gene. So um, it's going to give us the minimum fitness for any state, the maximum fitness for all states, and then the value that the gene needs to achieve the maximum state. Okay, so that's what the fitness mapping looks like, and that's for each implicit locus. And so the total fitness is simply the average of your fitness across your entire genome. So the environment switches, they simply alter what these mappings are. And so here's the code for the environment. So we have an environment ID for tracking purposes. We have a pointer to the implicit genome this is referencing, and then we just have a map of each locus to its fitness data, and the fitness data just has an optimal value, the minimum fitness, and the maximum fitness. So um, for example, if you have a discrete um, a discrete locus that has 25 possible values, a fitness mapping might have a um, an optimal value of, say, a fitness of 2. No, yeah. So let's say it has an optimal value at 5. Okay, so then it has a minimum fitness of 2, or let's say minimum fitness of 0.1, a maximum fitness of 2. So basically, how close you are to that value will tell you um, what your fitness is. So the the optimal value is where the maximum fitness occurs, and depending on how far away from that, you'll get closer and closer to the minimum fitness. So other implementation details. So as we mentioned, the fitness of an organism is calculated as the average fitness of its genes. So second thing is to make the simulation faster, we keep the population to a minimum. You can set this. It's the max orgs parameter. We default it to 10,000. So the way this works is, so if you imagine, let's say half your population has a fitness of 1. Let's say you have 10,000 organisms, half of them have a fitness of 1, half of them have a fitness of 2. So the next generation, you'll have the 5,000 that has a fitness of 1 will generate 5,000, and the 5,000 that has a fitness of 2 will generate 10,000. So you now have 15,000 organisms, and so it will randomly select 10,000 of those, and the random selection will maintain the general population dynamics, but the maximum ceiling will keep the processor from dying. So the mutation rate is per locus, but we're actually keeping stats per organism. So the beneficial deleterious ratio is not on individual mutations, it's on organisms as a whole. And the reason for that is because a lot of the mutation studies that are done with organisms are actually done around reproduction. And so those are kind of whole organism studies. So we're trying to mimic that. So we do set some arbitrary values. The maximum fitness of a gene is 5. Um, The maximum change on a continuous change gene is 0.25, and then the maximum number of states for a fixed configuration gene is 50. I don't think any of those dramatically impact it too much, but those are good things to keep in mind. So that's kind of where the simulation stands at the moment. Going forward, we've got several things we're looking at. We're trying to figure out what are the relative importance of various parameters. We're going to try to determine which parameters govern steady state behavior versus which parameters determine the environment switch behaviors. And then what we want to do is we want to kind of match those parameters to known organisms and compare and see if this simulator provides a first order approximation of what is actually happening in biology. Now, one thing to note is that when we talk about a locus in this simulator, it's actually slightly different from a genetic locus in an actual organism, because this is really a locus here is kind of a mutable site, not necessarily a specific gene. So if a gene has two mutable sites, then that would basically be two loci on the simulator. The other thing we want to do is right now the simulator is entirely asexual. It'd be interesting to model sexually reproducing organisms and see how that changes the dynamics. There's been some interesting studies where you have kind of isolated populations of yeast that they've brought together, and so it'd be interesting to model those dynamics with this type of a simulator. So anyway, looking for collaborators. So if this is something that interests you, take a look at the GitHub page, check out the code for yourself, run some simulations, and get in touch with me. And I'd love to talk about it some more. Or if you're more on the biology side and you want to compare how these results match actual biology, I'd love to talk with you and maybe get some collaborations going. So thank you very much. I hope this helps out.